All right, let's start talking about our second line of defense. We're going to break out the second line of defense into several different mechanisms that we'll talk about individually. So we have phagocytosis, inflammation, fever, and antimicrobial products, which are all part of this second line of defense. So let's talk about phagocytosis first. Phagocytosis comes from the Greek, meaning to eat a cell. So that CYT refers to a cell, so it's cellular eating. It's basically the ingestion of any microorganism or other substance by a particular cell. So phagocytosis can be involved in clearing away debris, such as dead cells, denatured proteins. So it's basically part of the second line of defense because it helps to ingest and get rid of some of these pathogens. It can also be used to, as it says, extract antigens. So some of these cells that interact with these pathogens will kind of spit out pieces of the pathogen, and they will be used as antigens. Right? They're kind of just components of important in the immune response. So any cell that can go through this process of phagocytosis is called a phagocyte. So once again, that's CYT referring to a cell. So phagocyte is a cell that goes through phagocytosis. All of them are white blood cells that we've kind of mentioned previously. So we have the neutrophils. So this is 40 to 60% of our white blood cells are neutrophils. We recognize that these were granular white blood cells. So they were the granulocytes. They had the dark granules inside of them when you stain them. And these granules can also be kind of released by the cell to kill other cells. But in releasing those granules, a lot of times they're killing themselves. The neutrophil kills itself at the same time. So neutrophils tend to be highly phagocytic. They're motile, they move around, they're active in the initial stages of an infection. They leave the blood and they go right to the infective site, and they're there to destroy the microbes. Neutrophils only live for a few days. And they tend to accumulate at the infection site. They're kind of your first line of defense, the first little warriors that get there to combat the pathogen. So a lot of times their little dead bodies tend to pile up in the pus that is produced at an infection or a site of infection. So a lot of times pus is, the main component of pus is all these neutrophils. Another type of phagocyte are monocytes. So monocytes are not phagocytic until they leave the bloodstream. And once they enter the body's tissue, then they mature into what we call macrophages. Right? So we saw earlier that the monocytes can develop into macrophages. And so once they become a macrophage, or in this process of becoming a macrophage, they actually increase in size and they develop these lysosomes, which are an important part of the phagocytic process. So the macrophages are there mainly to eat and swallow stuff that we don't want around. So they'll eat our old cells that have died, as well as functioning as part of our immune system. Macrophages tend to be much longer lived than neutrophils. They can live for several months. They can hang out in certain areas of your body, and they are garbage men, right? People describe them as kind of aggressive janitors because they clean up areas, and they can swallow up to 100 bacteria develop into that macrophage. So they're the garbage men. They're out there. They're kind of cleaning things up, getting rid of dead stuff, and they hang out for a long period of time. They know what to eat, right? So once again, these guys need to be able to differentiate between cells of your body and foreign cells. So they need something to say, hey, I'm a foreign cell, I'm different than your body. And so typically they're looking for some type of sign or a marker. So these markers are different types of antigens. They can be proteins, they can be sugars, they can be combinations of both. And the markers that many pathogens have in common, so kind of a common set of markers, are called PAMPs, or pathogen-associated molecular patterns. So PAMP, pathogen-associated molecular patterns. PAMPs are not found in mammals, so we don't have them. Good, because this is what our cells are using to recognize something that is foreign. So it's foreign if it has a PAMP. Now, our cells have receptors for the PAMPs. All right, so the phagocytic cells, the phagocytes, they have receptors that the PAMPs bind to. And these receptors are called PERs, or pattern recognition receptors. So the PER, but that's the way it goes. So our cells have the PERs, not the PAMPs. The PAMPs are on the foreign cells. So an example of this are what are called inflammasomes. So inflammasomes are found in the cytoplasm of these phagocytes, and they're protein complexes that are used to detect PAMPs. And when they detect the PAMP, they release a signal that triggers inflammation. Okay. Now, let's talk a little bit more about this process of phagocytosis. How exactly does it occur? All right. So here's the steps, and I'm going to show you an image in a moment of each of these steps. So I'll kind of briefly go through them right now. So we got chemotaxis is the first step, being attracted to that foreign cell. Adhesion, all right, so we have binding that's going to occur. Phagosome formation, kind of taking in the pathogen. Phagolysosome formation, starting to degrade it, and the destruction of the pathogen by enzymes, and then finally eliminating it from the phagocyte. So this circle above is showing you kind of looking at everything that's going on. So in purple is the membrane that's surrounding the eukaryotic cell, and then we've got these kind of rod pink bacteria here. So if this is our phagocyte, right, and this is our foreign pathogen here that we want to get rid of. So now we go down to the blown up image, and once again, the purple line represents our membrane around our phagocyte. We see these kind of Y-shaped receptors. These are the pattern recognition receptors, or the PER, that is on the outside. These are the little things sticking out all over the surface of the bacterial cell. So we said the PAMPs, right? So they're found on the pathogens. So the PAMP will bind to the PER, right? First, we need to have chemotaxis, right? So chemotaxis is this chemical attraction. So there's something about that pathogen, something that's being released from the pathogen, that's going to cue the phagocyte to kind of move to that area. So there's all kinds of different chemotactic chemicals that attract phagocytes. Some of them are components of other white blood cells or damaged tissue cells. So we talked about the cytokines. that can be released from other white blood cells that can attract other phagocytes to that area. So a lot of times having being released, that will be that chemotactic response. Okay, and then we have adherence, right, where the PAMP binds to the PER, and then that's going to trigger the membrane to invaginate here. So we see the membrane, the inside of the cell. These two sides of the membrane will fuse together, and this will form a phagosome. So phagosome, and S-O-M-E refers to a body. So this is a body or a vacuole or a vesicle, whatever you want to call it, that's inside of the phagocyte right now. Area inside of it, bound vesicle. So we've taken the bacteria inside of the cell. These lysosomes, which are organelles that are inside of our phagocyte, and they contain digestive enzymes. So the lysosome will move over the phagosome, and it will fuse the membrane. The lysosome's membrane will fuse to the phagosome membrane. The enzymes that are inside of the lysosome to flow. Fusion of the lysosome and the phagosome is called a phagolysosome. The whole body is called a phagolysosome. And now we have the digestive enzymes inside the phagolysosome. And as you can see, these consist of lysozymes, DNAases, RNAases, proteases. So these are all kinds of enzymes that are going to break proteins and basically just destroy the bacterial cell. And then whatever is left over, that phagolysosome can move up to the outer membrane. The phagolysosome will fuse with the outer membrane and expel the contents to the outside of the cell. And this is the whole process of phagocytosis, of kind of bringing in that pathogen and digesting the pathogen, completely killing it off. And that's the role of these phagocytic